Yeah, a very good evening to all of you. I am Dr. Dharmendra from Sanskaram University, Jhajjar, Haryana. Today I present role of natural polymers and drug delivery systems. As we all of know, so many types of drug delivery systems are available in the markets like solids, liquids, semi-solids, aerosols in form of gases. But nowadays, the pharmacokinetic parameters of this drug may not be suitable for the patient to give the potential therapeutic or clinical effects. So nowadays, scientists need to find out some better options to give the drug as a good delivery systems with the best pharmacokinetic parameters. So in this continuation, I am isolated a new best pharmacokinetic polymers. This give the best biocompatibility, biodegradability, non-toxic stability with the drugs. So as all of you know, so many types of pharmacokinetic parameters and the polymers are present in the in this scientific world. So today I am present about the natural polymers. Natural polymers means the polymers those are come from the natural source and their types and different source of the natural polymers. And I highlighted the role of starch in the delivery systems because starch have the some ideal and very advanced contents compositions to give the delivery of drug in the patients or in the pharmaceutical science and the scope of the natural polymers in advanced drug delivery systems. So natural polymers, all of you know, are the composition and repeating units of monomers. These may be linked with the covalent bonds and the living or produced from the living organisms. So many types of natural polymers are present in the era like cellulose, starch, protein, DNA, RNA, chitin. But I highlighted the starch in my research. Because cellulose are found in the cell wall of plants, and these are the polysaccharides composition. These are the abundant organic polymer on the earth, and starch also we come under the polysaccharides, or it is the composition of glucose. But the arrangement of these glucose in a very well maintained methods. These are the two types of glucose: amylose, amylopectins, and amylose are the sour branch glucose, and amylopectins are the very branch amylopectins. So I observe and I identify the role of amylose and amylopectin in the starch as a drug delivery systems. So there are the other starch also present as the proteins, DNA, RNA, chitins. All these starch can be used and used by the various scientists as the drug delivery systems. But in my research, starch was selected as the drug carrier because it gives the very good pharmacokinetic parameters for drug delivery systems. So, or more deeply, I have isolated starch from banana. So the name of this starch is the banana starch polymers. The theory behind the isolate of this specific banana starch because of the containing the specific composition of amylose and the amylopectins. Because amylose is the 26 to 34%, it's maybe based on the geographical locations, and the amylopectin is 66.74%. And the amylopectin composition is the area of research that interests me to formulate the new drug delivery systems because amylose is a short D-glucose linked polymers while amylopectin has a branch and polymers. So amylose give the immediate release for the drugs and amylopectin give the sustained release. So this is only the starch, or this is only the natural polymers, or a single polymer give the dual release patterns as the immediate and the sustained. So many types of formulations are available in the markets. Those give the immediate and sustained release, but a single polymer give the both or the door release. As my knowledge, there is the first polymers or the first natural polymers that perform such type of properties. So in my research, I prepared the nanoparticles of this banana starch by using the nanoprecipitation methods. All of you know the nanoprecipitation methods depend on the precipitation of nanoparticles. So I precipitated the nanoparticles based on the acidic and basic solutions because my starch was now dissolved in the water. And so I formulate and I composed a new basic solution that composed the NaH, urea, and H2. So that basicity enhance the solubility of the starch, and I add my isolated compounds or API or active drugs in the basic solutions. And the another acidic solutions that I use in the 0.1 normal HCl, the solution are because of the precipitation to real the precipitations, and then centrifuge and lyophilize the nanoparticles to collect and further uses. So after sorry, to before going to the formulation of nanoparticles, I'm also used the compatibility study of the banana starch and the drug that I have used during my research. So my drug was the quercetin, so I will check the banana starch and quercetin are compatible to form it in any formulation or compositions. So there was no any other peak was found. So it FTR study confirmed that these both banana starch and my drug are compatible to form a new formulations. And then I tested the entrapment and drug loadings of my formulations. So where in the first times I was found very good drug loading and entrapments because my drug loading was 51.9% and the drug entrapment was 41.50%. And after that, I have judged the particle size of my formulations. So it was a very good particle size. It was a 76.89 Z average of my formulated nanoparticles of banana starch and the drugs. And I have also searched the shape of these nanoparticles. So shape of these nanoparticles are like spherical and very good in the size and the shapes. So I was also going to evaluate the properties of these prepared nanoparticles as in vitro drug lineage. I have formulated so many formulations, but any of the formulations, my four best formulations, is named as BSN1, BSN2, BSN3, and BSN4 that give the good release of drug as immediate and sustained release. As in the form of immediate release, my BSN1 give the 44.8% and BSN2 42.52% and image 3 30.75% and 34%. And that immediate release was resulted because of the amylose composition in the starch, and the sustained release that was 96.96%, 96.83%, 97.85%, 98%. These all release are responsible because of the amylopectins or branched sugar molecules within the 12 hours of release, because the entrapped drug while the BCS class 4 drug and BCS class 4 drug have the low solubility and low permeability. So the release, so the drug delivery system for such type of drug or BCS class 4 drugs is need to give a specific drug delivery system that can be penetrated in the skin by using the size and should be soluble because of the reduction in the size. So I formulated this formulation with both the pharmacokinetic parameters, solubility and sustainabilities. So this is the cumulative drug release of my formulations, and I'm also judging the antioxidant activity of that formulations in different volume and different concentrations. So with the standard that is the free quercetins that I use as the standard drugs. So my formulation VSN Q1 was found to be more effective than the standard drugs. So such type of results agree me to if my formulation is antioxidant, it may be the anti-cancer activity. So before going to anti-cancer activity, I'll check the cytotoxicity study of the prepared nanoparticles. And the cytotoxicity study was done by the MTTS against the NCL-S66 cancer cell lines. And the result of this study is also very good. And it gives the more than 86% of uh, susceptibility to kill the cancer cancer cells in the cancer cell lines. So after that, I am also check in vitro in vivo drug studies at the wound healing. So wound healing contexts of the prepared formulations are found to be very good in case of QVSN1. That was QSN1 was selected for the in vivo drug strategy because it was the good in the particle size and in vitro drug release. So I select the QS12, the antibody contractions. So when I judge the histopathology of healed tissues of the albino star rats, I will found that the growth of fibroblast, regeneration of dermis, inflammation cells are reduced, hair follicle was found, fibroblasts were found, blood cells were found, macrovasins. So the healed wound skin give me the positive results also. So this formulation was also found to be very good anti-wound healing formulations as a new drug delivery systems. So after that, I am also check the anti-inflammatory activity of my formulations. So anti-inflammatory activity of formulations after to check the percentage edema inhibitions. I am also the histopathologies that give the more clear results how my formulation is works. So the cross sections of this histopathology give me the ideas how the inflammation was, how my formulated drug give me the results as it increase the epithelial tensile and also come in the same age the previous before the inflammations. So such type of results also prove that this drug delivery system, this new starts that give a very good particle size that is 76.89 nanometers, and it also give the good drug entrapment that is 51.54 meters, and it's give the good drug entrapment that is 41%. It also give the dual drug release that is the immediate release at the 45 to 46% within the 2 hours and 96 to 98% within the 12 hours. So this only the single starch, single nanopolymers, that is the starch, or specifically the banana starch, because only the banana in the category of starch, only banana starch contain the 26 to 34% of amylose and 60 to 74% of amylopectins. So this dual release was found because of this composition of amylose and amylopectins. So this is the summary of my results that I have already discussed, and these are the publication of my research at healing and extent potential of QVH published in the AIMC. That is the Bentham Science Scopus paper, and the another formulation of self-pressure and starch particles was published in the Nanoscience and Technology here. It is also national Scopus, and I have filed Indian patents bearing the number as given by screens. So thank you. This is from my side.